So good evening, everybody. I hope you can see my slides okay. Calling in from the UK at the moment. It's about 2 o'clock, so I apologize if it's a little sleepy. But today I'm going to talk to you about some of the work that I've been doing in my PhD. So this is in collaboration with Roche and with the Kennedy at Oxford. So this audience kind of needs no introduction with this, but I'll give a brief one. So we know that the microbiome really drives a number of host processes in a rather multifaceted way. So I've used macrophages as an example here, but in immunity, we know that colon-derived butyrate from the microbiome there, which we found in the Paris lab a couple of years ago, as well as lactate from the Ileal microbiota, really drive different macrophage subsets. So whether they be intestinal macrophage subsets or kind of tissue-resident liver macrophage subsets, and they drive them towards an antimicrobial profile. And it's not just this kind of microbial control is not just limited to immune processes. So there's been recent data coming out showing that we are looking at this change of lipid metabolism when we assess germ-free mice right the way up to SPF. There's a variation in stool lipid content, in weight, and it's all kind of starting to be linked with the microbiome. So if we narrow this in, we know that the host and microbiome communicate in the small intestine. So a good example of this is bile acid metabolism. So bile acids travel through the small intestine, helping in dietary metabolism, and then are recirculated. 95% of them are recirculated back through to the liver. But there are other microbe-mediated processes that happen there, as well as kind of shunting on to the large intestine. But what we're unclear on is how these microbial metabolites that are present in the ileum interact with the host and affect its gene expression in kind of a dependent manner. And there are some current ways we can use to analyze this. So metabolomics, that would get the host input. So you're really not distilling out the microbial impact. And metagenomics, that correlates with what's happening in the host, but just because you know something's there doesn't mean you know what it's doing. And you might be thinking, Amit, you're talking about the microbiome. Why are we focusing on the small intestine? And I love bringing up this figure. This is from the CAPSCAN paper last year. And you can really see that there's a diversity of metabolites that's not really captured at all when you're looking at stool metabolomics. And this is from humans. You can see a really large diversity, which we don't often get. So it might reveal some additional crosstalk that we're often missing. So to do this, I borrowed a technique from synthetic biology. Now, I appreciate Professor Givens gave a very, very rounded overview of this, so I'll be brief with this. But I used Metagenome-scale metabolic modeling. And to summarize, it's taking effectively what is a list of reactions and using flux balance analysis to plot the path of metabolite through this chain of reactions, optimizing for a particular objective. So again, we know that microbes tend to optimize for maximizing biomass. So that's the one that we used here. And again, we can scale this to a whole microbiome. So making the assumptions that all the cells are at steady state, everything is kind of well optimized. And this kind of is incorporated in this workflow. So we didn't initially use a whole SPF microbiome. We started smaller. We used the MM12. So this is a synthetic 12-member consortia that's really designed to recapitulate the functions of the colon and fight against colonization resistance. There isn't an Ileal synthetic microbiota yet that I know of. So this will have to do for now. And we took 14 12-week-old female mice. They were all fed the same diet and taken down around the same time, about 2 p.m. And then there were 2 work streams that happened from there on. So we did the microbiome work stream, where we did the qPCRs on the 16S regions. We took Agora-2 gems. So these are very well-defined genome-scale metabolic models from the Teal lab in Galway, and a custom mouse diet. And the real difference between some of the work in the human side is we tend to know what people eat, and you wouldn't believe how difficult it is to figure out what is in a mouse diet. There's not a lot published out there so far. And then I actually ran it through Mecom. So this was the tool, again, that was explained previously, to kind of process what was going on in the microbiome. And what we were looking for as an output was net metabolite efflux. So what was the microbiome just effluxing out into the system? On the other side, we had some Ileal RNA-seq data. So I took this and did differential gene expression versus a paired germ-free cohort. And we wanted to understand what microbe-controlled genes were present there. And then finally, we brought these together and did some Pearson's correlations to really see where those metabolite and genes kind of lined up. So first, looking at the host. So we saw that there were over 1,200 Ileal MN12 control genes, and these seem to largely dominate the immune phenotype, which was upregulated. And this is really consistent with the literature, saying that generally germ-free mice don't have as strong of an immune phenotype. And we saw that there were these kind of glycoproteins, cell cycle type genes, which were not recovered from the germ-free phenotype. So again, really focusing on this immune subset. Then kind of shifting focus over to our abundance data, we saw this dichotomous phenotype appear. Now, perfectly honestly, we don't have an explanation as to why this occurred yet, but it appears that there was really two kind of major players there. One was this real domination of Lactobacillus reuteri in our in this kind of low Shannon index group, and the other was we saw this generally Akkermansia kind of forward phenotype in our higher Shannon index group. This data is kind of consistent across what we've seen at the Kennedy and other mouse work there, and there is some emerging evidence to say that this might be a facility-specific thing, but this is something we're just going to keep in mind moving forward. And when we look at this a bit further, when we're looking at the PCOA clusters, we can see that, again, our over 80% of the variant is driven very much by this Lactobacillus reuteri dominant samples. We did try and have a look at whether the cage had an effect in there, and it was unclear whether it really did or not. So again, something we're just keeping in mind. Next, we looked at whether what the Ileal MN12 were producing, and we saw this really wide, diverse range of metabolites that were coming out. So very in line with what we're kind of expecting from usual colon metabolic outputs. And these, again, split into their two diverse groups. So the Shannon index low ones were very much dominated by D-lactate production, and some of the others were looking at more at like this. There's a high proton phenotype, again carbon dioxide. So there's different things kind of being produced by different groups here. And then we took this a little bit further and looked at how our samples separated based on PCA, and we can see again lactobacilli really kind of brought out this cluster here. Akkermansia brought out another cluster. So it's showing that these metabolites are actually mapping onto what we expect in terms of abundance. Okay, so finally we had a look at whether these predicted metabolites generated anything that correlated with gene expression, and we did see some interesting things come out. In particular, I'm interested in this ARID3A gene, which correlated with some pyruvate, D-lactate. ARID3A is already a kind of well-established gut-liver gene, actually. So this correlates with colorectal cancer, as well as intestinal cell motility. And there's some other interesting immune-type genes that were coming out, so the granzymes. And we took this a step further, and we had a look, is the diversity linked with this? At first glance, we couldn't really tell. So there's some which were quite linked with this. So you can see that the samples with quite a low Shannon diversity tended to go down in this carbonic anhydrase gene, but it's not consistent. So I think what we need is a few more data points to kind of really push this home. These bottom panels, though, kind of start to have a look at where we could generate some hypotheses, though. So looking at this insulin-like sensor, this is really linked with hepatic glucose production. And this MARX SL1 is actually quite linked with M cells. So M cells are often found in gut-associated lymphoid tissue, so the Peyer's patches. So this is really interesting that this is linked with acetate. And finally, we've seen this Margit1 linked with ammonium. So again, this is linked back to lipid metabolism, so all processes that occur in the liver, in the ileum, sorry. So it's something we can kind of take forward and investigate further. So what have we learned so far on this? So the ileal microbiota in human-12 is kind of largely dichotomous in our phenotype, and that metagenome-scale metabolic modeling provides this unique way to capture this functional complexity. And finally, correlating it back to host transcriptomics provides us functional insight into this host microbe crosstalk. So to take this forward, we have some SBF scale samples, which both have young mice, like I showed you today, as well as some older mice to see how this dynamic changes in the aged phenotype. And then we want to go back into the lab and do some direct gene perturbation to see, is what we've seen actually valid? Is there a real phenotype we've seen here? I'd like to thank all the people that have helped me work on this, Dr. Nicholas Ileal, Professor Fiona Parry, and Dr. Petar Skamanovich, my supervisors. Thank you very much for listening.